Yeah, good evening. That what are microorganisms? What are microorganisms or what are microbes? What are they? Having known what microorganisms are, which you know very well that if we are talking of microbes, we are referring to the certain set of microorganisms or organisms that are too small to be seen with naked eye. And classes of such include viruses, bacteria, bacteria, sorry, bacteria, parasites, and fungi. Sorry, this is better to be protozoal. We are talking of viruses. Viruses, example of viruses include CMV, that is tobacco mosaic, coronavirus, then example of this include the E. coli, you know very well of this. An example of a protozoa is a Plasmodium species. Plasmodium species. Okay. Fungi. Fungi are a group of organisms that belong to the group Mycota. So an example of this is our normal yeast and other. Okay, so what are microorganisms or what are microbes? Then all the traits, that is, if we look at a typical microorganism like this, that means, and let's say this one is a virulent trait of a microorganism. Let's say this is microorganism. In this microorganism, for example, if this one is a E. coli, and it is generally referred to as if it is pathogenic bacteria, So what actually determines the degree of infectivity, the degree at which this microorganism will cause an havoc, is known as its pathogenicity or its virulence. So, and in the one of my videos where I mentioned in immunology, where I talk about the interaction between host and pathogen, so should just be that the host will be looking for a mechanism that's going to make it be able to be better adapted into its environment. So if we are talking of E. coli, for example, now the virulence gene, that means pathogenic organism must contain certain genes that will make them to be virulent, that will make them to be deadly. So, and these genes, the question is, how is it how is it being expressed? How is it expressed? That is, there must be certain level of communication or what we can call interaction between the host and the pathogen. Before any disease or infection will be initiated, that means a pathogen must send a kind of signal to its host. And it is this signal that we call virulence gene. So this virulence gene, how is it being expressed in microorganism or in certain bacteria? How is it being expressed? So we want to know. That means there must be a kind of communication between the host and the environment. Now let's say this is the pathogen DNA or the hereditary material. If this is the pathogen DNA, now let's say on this DNA there are located. That's why we say that genes are certain segments of DNA. They are genes that are located variously on DNA. So it could be that, let's say this one is A, B, C. So the segment A, B, C could be the one that is helping or making this organism to be virulent. So the question is, how is it being expressed? Don't forget that if we are talking about E. coli, the lifespan of E. coli divides every 20 seconds. Every 20 minutes. So that is telling you that if you have one E. coli, at the end of 20 minutes, this E. coli must have undergone binary fission, and that binary fission okay, so this one, as I said, must have, this is at the end of every 20 minutes. If this is the chromosome of the DNA of E. coli, and the one that is indicated, the segment that is indicated in black color, let's say it is called section ABC, which I said, let's assume that is the virulent genes of this microorganism. So immediately this one wants to undergo diffusion. The first thing that is going to happen is just that the first thing that is going to happen is that, let's say, so this microorganism first of all will undergo what is called replication. It's going to duplicate. It's going to duplicate itself. It's going to duplicate the DNA here. So it's going to be like this. Okay. Let's say this is if this is the ABC. So this one will undergo duplication. That is, it's going to be like this. So this one is going to be replication. So that at the end of the day, this one will undergo further replication. So this one will now become 4 after second generation. So it is expected. Don't forget that every organism must look for a way of adapting to its environment. That is, must be able to know how they will be able to adapt to their new environment, and that one is going to help them. So what we are saying in essence is that if this one is the virulent genes, it is the primary aim of this organism is to make sure that it propagates the virulent gene for certain number of period, for certain number of generation. That means that gene must be a functional gene. The gene for that virulence must be functional gene, and it must be maintained for a certain period of time, long period of time, for this microorganism to be able to retain is a virulence. And don't forget that I told you that it is the primary aim of every microorganism, of any organism, to make sure that it replicates its most important genes to be able to adapt into this new environment. Now, what microbial genetics actually involve is that how are important genes, or what you can call important information, how are they expressed, the expressivity, how are they expressed over a long period of time. So that means that kind of genes must be duplicated. It must be duplicated, and in trying to duplicate this, that means it must be able to be maintained for a very long period of time. Now, after knowing how are they expressed, the next thing is, how can they pass it from one parent to the offspring? How can they pass it from one parent to the offspring? Because what is going to be the essence of that microorganism that has actually acquired certain numbers of certain types of genes, and they are not able to pass it from parents to their offspring. So that is telling us that apart from the organism making sure that their very important information are expressed, they must also make sure that that kind of genes are also preserved through different types of generation. So we must know that. So it is studying of microbial genetics that is going to help us to know that. Don't forget that genetics is just the science of studying variation, heredity, and studying of genes. Don't forget that I told you that a gene is a certain segment of DNA, which must be coded for a very important and crucial gene for the survivability of microorganism. So in other words, in this course of microbial genetics, it is onus on us, or it is onus on all microorganisms to be able to express only their functional parts of genes. So, and they do this through the process called replication. There are other mechanisms of this that we are going to look at. Now, don't forget again that E. coli consists of around 4,600. It's only made up of around 4,600 genes. So, and not all these genes are preserved, or that can be preserved throughout their generation. There are certain ones that after replication, they will lose it along the line because these are non-functional genes. For example, don't forget the theory of Jean Lamarck about evolution, the theory of use and disuse. That is telling us that this E. coli will only be looking for a way of making sure that very functional genes, or out of these 4,600, are expressed continually, so that for them to be able to survive in their host, just like I said. Then human, that is in Homo sapiens, we have as much as between 20,000 and 25,000 genes. That is, human being contains just as like 25 times 5 of E. coli genes. Whereas in Mycobacterium tuberculosis, MTB, which has around 4.4 million. So, in the real sense of it, you will just think that E. coli contain as many as times 5. Human being contains as times 5 of E. coli. Now, how many 20,000 can we see from 4.5 million. That is telling us that if we want to look at it from that angle, we'll just try and see that maybe Mycobacterium tuberculosis is more complex than in human, because Mycobacterium tuberculosis, they have more genes than this. Not at all. That one is talking about. Don't forget that in prokaryotic, we don't have. They are not made of exon and introns. Okay, they don't have coding and non-coding. Then there is nothing like what is called processing. That is post-transcription processing. We are going to see all this because there is no way we will study microbial genetics without talking about how genes are expressed, how are they coded, and how can they pass from parent to the offspring. And that is the reason why the first thing that we're supposed to know immediately after knowing the composition, what make up a whole organism, and having known what make up a whole organism, how are these genes, how are they being replicated, and how can they be passed from their parents to the offspring. I have told you that human being is just almost times 5 as complex as that of E. coli. So, and that's the more reason why we will know that human being, although human being, because they contain more of functional genes than Mycobacterium tuberculosis, and that's the reason why that in prokaryotic cell we are not talking of post-transcriptional processing, which include like capping. We are going to see this as we go on. Don't forget again that in E. coli, or in my earlier video, I was able to differentiate between prokaryotic organisms and eukaryotic organism. And you know, whenever you talk about prokaryotic, that's the most primitive in their genetic makeup. And in eukaryotic, just like I told you, that one has more of introns than in prokaryotic organism. So our knowledge in microbial genetics will help us to know a lot of things in biology. For example, microbial genetics is exactly what the framework to be able to understand how complex a microorganism could be. And it is this knowledge of microbial genetics that we will always apply in practical situation. So for us to know more about the complex application or practicability of microbial genetics, especially when it comes to the industry. And if we are talking about application of microbial genetics in industry, that is when we'll be looking at the area of biotechnology, recombinant DNA technology. So it is part of the use of our knowledge in microbial genetics. Okay, then it is also microbial genetics is exactly what we also use in personalized medicine. So all this personalized medicine has been a recurring decimal in modern biology. Personalized medicine. So we are going to make use of this. And microbial genetics, the importance or the applicability is just coming into fold. Don't forget that everything in microbial genetics or in biology revolve around DNA, and that is the reason why that 1953 was a turnaround for a special part of a special part of biology when Watson and Crick's unravel the structure, the double helix structure of the hereditary material, which is the DNA. So now, the question now is how? The question now is how do genetic or how are genetic information being made of? How are they being made of? That is, what are the components of the genetic information? Now, a lot of time, we will still refer to. There was a time that there was a kind of controversy of which one is really the genetic material. Which one is really the genetic material? Because inside the nucleus of the genetic material of any organism, we have chromosome that is located inside that, we have DNA inside that, and we also have protein. So the question then was, which of these, could it be protein that could be the hereditary material, or could it be the DNA? So that was when that's what will lead us back into going historical in trying to know the history about microbial genetics and what is really in that genetic information that can be passed from parents to their offspring, and be able to make sure that vital genes by that microorganism is being retained for several generations. So that is why I want to start the historical background and date it back to the days of Frederick Griffith, which was in 1928. Frederick Griffith in 1928. It was this man that came up with a very simple experiment that actually attributed. He was the one that DNA, not the protein in the nucleus, that was the genetic material. That was the one that was put forth by Griffith. And this experiment will be further illustrated as we go on, whereby he was able to make use of certain strain of pneumococcus, certain strain of streptococcus pneumonia, certain strains of pneumococcus pneumonia. There are two types, basic type of this microorganism, of this bacteria. The first one is the one that has a smooth coated pneumococcus, and the other one is rough coated pneumococcus. So smooth coated pneumococcus and the rough coated pneumococcus. This one, the smooth, was found to be non-pathogenic. It was found to be non-pathogenic, and the rough coated one was found to be non-pathogenic. This one was a kind of it was non-pathogenic, that is, it was non-virulent. Then, if it is non-virulent, that means it cannot pass its hereditary material from the parent to the offspring. And just like I said, it was this man that first nailed it on the head that it was a DNA, that DNA is actually the genetic material, not protein. So immediately after this, some other scientists came around in 1944, and that was Avery, the following scientists, Avery, um, MacLeod, 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 and McCarthy, and McCarthy. All these in 1944 were able to know the exact plant, I mean the exact microorganism, the exact material that was actually responsible, or this what I will call the mechanism of genetic transfer. And that was when they came about what is now known as a transformation principle. Transformation principle. So all these could be dated back to the background history of this. Don't forget that I may not be able to go into detail of these experiments, because it is a kind of revisiting the milestone and the historical background of how microbial genetics has actually gained ground in our modern biology up to today. So now, just like I told you, I've asked this question. That's the first question, or the first puzzle that we are much concerned with, is that number one, what are the components of the genetic material? What are the various components of the genetic material? What will and how will microorganisms, how will they pass their most essential genes? How can they pass it to their generation? Knowing fully that, just like I told you, that the lifespan of E. coli, regeneration time for E. coli is always every 20 minutes. That means if there is any mechanism that will not allow E. coli to be able to propagate every 20 seconds and 20 minutes, that means it's not able to fulfill the theory of genetics, that is microbial genetics. Don't forget that I told you genetics is just the ability of an organism, or the study of the mechanism of an organism, to be able to pass certain functional genes or genetic information from themselves, which happen to be the parents, to the next generation. So now, after 1944, the next milestone that we will never forget, just like I told you, that was in 1953. And that was when Watson and Crick were able to actually come up with the exact structure of the genetic material. Don't forget that I told you that 1928, the establishment of which one was the actual genetic material, was it DNA or the protein? So, and I told you that Griffith came up with his own experiment and was able to lay every other controversy to rest, and affirm that DNA was the exact genetic material. So after that, the next thing was in 1944, when other people like Avery Chase and McCarthy and all the other scientists now work on the work of Griffith of 1928, and they were able to also talk about transformation principle. Okay. Now, 1953, I said that was when the actual structure of that DNA, which happened to be the genetic material, the actual structure was discovered by Watson and Crick. And it was after this that more knowledge actually came to know that there are three different types of DNA. It was according to that we have X DNA, we have Y DNA and we have Z DNA, and depending on the prevailing environmental condition, that's going to tell us at which stage will that DNA be. Is it going to be at a Y stage? Is it going to be X stage, or is it going to be Z stage? But we have to know that between this, which is called the minor groove, we have between 10 and 11 base pairing or base pairs in that. Don't forget that DNA contains cytosine, adenine, thymine, and guanine. These are the nucleotide bases that we have in DNA. So immediately after 1953, the next thing was in 1956, 1961. What happened in 1961 was that Francois Jacob was able to describe what is now known as the lac operon. The lac operon. So we are going to see more of this in lac operon, because lac operon will actually tell us the way how do microorganisms express their genes. I have told you that they have a way of doing that. Then the next one is operon. They now have operon, that these are the set of genes or the arrangement of genes on the microorganism that they are able to use it for regulation of expression of their genes. Now, the next thing that we also need to know very well in this course is the theory of central dogma theory. What they call the central dogma theory is very, very important. That is, DNA, which we now know as the genetic material of microorganism now, will be transcribed. I told you we undergo what is called replication. If it undergo replication, this replication will now be transcribed into messenger RNA. Genes are stored in form of messenger RNA. Now the set of genes that are now stored in form of messenger RNA will now be translated. That is, it's going to be translated into amino acids. And don't forget that we have 20 major amino acids, and it is these 20 amino acids, that will organize themselves into thousands and will now be able to form what is called a protein. So this is exactly what we known as the central dogma theory. So in this course, it is very, very pertinent that we must understand very, very well how genes are expressed, what are the genetic makeup of individual cell, genetic inheritance, genetic substance, and after that we are able to look at the historical background of this. Now let's now try and familiarize ourselves or get acquainted with some of the terms and methodologies, terms and terminologies that we have or we mostly use in microbial genetics. The first one we'll consider is the genome structure. The genome structure. This is very, very important. So the microbial genome structure, just like I have showed you before, they are simpler in prokaryotic cell, that is in microbial cell, than in eukaryotic cell. They are simpler in prokaryotic cell than in eukaryotic cell. So after that, we will also come across what is known as a plasmid. What do we know? What do we have to say about plasmid? Plasmid are just small. They are small, very, very small, and also circular. They are small and very circular DNA, which are extrachromosomal in nature, and the organism that possess plasmid means that that plasmid is going to be very, very important for them for survival. So any microorganism that possess plasmid means that that plasmid is going to confer a special adaptability on them than any microbial cells that do not have it. So it is very, very, so they know that they normally use that plasmid. You know, I told you that it's always circular, very, very circular and very small. So this one confer extra stability. It confer extra stability, extra virulence. In fact, that is exactly what microorganisms use for resistance, antibiotic resistance. So that is telling you that if you see any strain of microorganism, one has plasmid and the other one doesn't have it, that is actually telling you that the one that do not have it will be sensitive, will be easily damaged, or will be easily controlled by common antibiotics. Whereas that one that have that plasmid, that's telling you that it is going to confer special stability to that, and that is what is responsible for drug resistance. So apart from that, I told you also that one other thing that we must know is that how are genes expressed? How are they expressed? Then not only that, how are these genes, how are they regulated? If genes are expressed continually, if genes are continually being expressed and there's no way of regulating them, that means there will be problem. And that is the reason why this man of 1956, Jacob, 1961, actually talked about lac operon. That is, these are set of genes that have the ability to be switched on and the ability to be switched off whenever they have, all these important genes have been expressed. So we need to also get acquainted with that. Then another thing is, I told you that microbial genetics has actually helped us to talk about what is called genetic engineering. There's no way we will talk about microbial genetics without talking about genetic engineering. And what's another word for genetic engineering? That is recombinant DNA technology. Recombinant DNA technology. So we are going to know about this. Then in this course as well, we will also come across what is called diversity and adaptability. Diversity and adaptability. It is the diversity. Don't forget that the characteristics or the genetic makeup of any individual, be it microorganism, be it eukaryotic organism, is not going to be 100% the same as after they must have undergone transcription, translation, or replication. So we will actually see that as well. So it is this diversity that actually determines their method of adaptability. Then we also come across mutation. What are the factors that is actually responsible for mutation? And what do we mean by mutation? And that is the sudden change in the genetic makeup of a microorganism. The sudden change in that. We will also look into that. Then there's no way we will talk about microbial genetics without looking at evolution. I told you that as a student of biological sciences, we have to get ourselves acquainted with theory of evolution in all organisms, not only in human, in also organisms. microorganisms. Sorry. So microbial genetics. So we want to, something might be telling us as what is or what are the relationship between genetics and microbial genetics, between genetics and microbial genetics. Don't forget that in broader sense of it, it is genetics that actually laid the foundation for microbial genetics. So, and that is the reason why we talk more of gene expression. We highlight some unique aspect of microbial genetics or microbial expression. We highlight certain like DNA replication, transcription, translation, protein synthesis, mutation, diversity, and others. So, and just like I told you, that microbial genetics is a branch of genetics. Genetics is the universal set, whereas microbial genetics is just a kind of subset under that. So to be able to understand microbial genetics, there's no way we have to divorce genetics from microbial genetics. So now, before we go on, we have to look at, I've mentioned that we have to look at DNA as the hereditary material. DNA as the hereditary material. And it is from here that we are going to continue our next video from. And don't forget that in this video on this subject, we have been able to look at the introductory aspect of microbial genetics, whereby we are able to look at some very important terms and terminologies in microbial genetics. We are able to look at the central dogma theory. We also made mention about the principle of transformation, or the principle of genetic diversity. Then after that, we were able to look at what plasmids are, and I told you that plasmids confer a special character or special on microorganism, because it is this plasmid that happen to be circular, very, very small, that they normally use for very important mechanisms such as express the genes that will be responsible for virulence, and that will be the gene that is going to make that organism to be able to survive or to be able to adapt into the new environment. And that is what is very, very important in this microbial genetics. Don't forget that today is just an introduction to microbial genetics. We are going to continue from there. And if you also need this slide, please comment that in the comment section by putting down your email, and subscribe to this channel as well. Thank you.